I'm Ashok Venkatraman, and I'm speaking to you from Singapore. And it's a great pleasure to introduce the panelists who today are Professor Bill Kalin from the Dana-Farber Cancer Institute at Harvard University, Sir Peter Ratcliffe from the University of Oxford and the Francis Crick Institute in London, and Sir David Lane, who is the Chief Scientist at A*STAR in Singapore. I understand that Peter and David are joining us from the UK, and Bill from a very early start in cold Boston in the United States. So welcome to all of you. It's such a pleasure to be talking to you this evening on a wide range of topics that really come from the work that all of you have done. The audience will be aware, Bill and Peter, along with Greg Semenza, shared the 2019 Nobel Prize for their discovery of the mechanism of oxygen sensing by cells and how cells respond to low oxygen tensions or hypoxia. And David is internationally recognized for his seminal work on the discovery of the P53 protein, which is a central sensor of stresses in cells and is responsible for suppressing the onset of cancer. So I hope that the audience, like me, is very excited to talk to the panel. Please do table your questions as the discussion goes on. They will be picked up by my colleagues, and if time permits, we will call on several of you to voice your questions to the audience. So with that by way of a prelude, let me start with asking the panelists to first comment on a question that I'd like to pose them to kick the discussion off. The way that cells sense oxygen and respond to low oxygen tensions sounds at first glance like a somewhat esoteric area of science. Peter, could you please start by telling us, followed by Bill and then David, about what it is that interested you about this area and why you feel it is so central to the way that organisms thrive?
Sure, Ashok. Well, of course, oxygen is of fundamental importance for all higher forms of life. It's the fuel that powers respiration, that creates energy. It's involved in countless biosynthetic detoxification and other reactions in the cell. And the fundamental problem that large animals, and that's what human beings have, is the delivery of oxygen in precisely the right amounts to 40 trillion cells or so. That's a huge physiological challenge. And of course, failure to do so properly generates hypoxia, and that's the complication of most human diseases. So I guess perhaps just briefly, I might just twist your question. Of course, lots of other people could have seen that before. So an interesting thing is why was this question lying around at that time for people like myself and Bill and Greg to pick up on? Why hasn't it been solved by others? And I think the reason is twofold. One, you must look at things at the right level, and people were trying to deduce mechanisms of oxygen sensing through observations on the whole animal, which were hopelessly confounded by heterogeneity of the response. And after many, many failed attempts, the field became unfashionable. And great credit should go to Franklin Bunn for opening the tissue culture models that enabled us to start. But also I think we're sort of slightly rebellious people who were happy to go into an unfashionable area, not one recommended by our mentors. So I'll stop there. Others may have other experience.
Thank you, Peter, for raising those interesting points about the philosophy of your science as well as the science itself. Bill, over to you.
Well, as Peter just said, of course, oxygen is fundamental to animal life on the planet for the reasons he stated. The only elaboration I would add is that I think most of the students are aware that too little oxygen creates problems, but less appreciated is the fact that too much oxygen can cause problems as well. So in the course of evolution, animals needed sort of a molecular rheostat to sort of get oxygen delivery right. And so it was very gratifying when this molecular pathway started to come into view to see this elegant mechanism, which of course is a tribute to nature, not a tribute to the scientists who were doing the work, and just how nature solved this problem of trying to get oxygen delivery right. And as Peter alluded to, not surprisingly, there are a number of human diseases where oxygen delivery is part of the problem, either too much oxygen or too little oxygen. And so, in fact, once we understood the mechanism in some detail, it created some translational opportunities that we'll probably talk about later. But I just want to echo Peter's comment, which is, in the end of the day, this was a triumph of hypothesis testing, reductionist biology. Let's just figure out how things work first, and we'll worry later in terms of whether there are any translational implications. I think today there's too much emphasis on tell me why what you're working on is important, why it's going to be applied in the clinic. I think you have to do the so-called blocking and tackling first, understand the system, generate some knowledge, see what you found, and then maybe once in a while, if you're lucky, that will lead to some clinical opportunities.
Thank you, Bill. That was, again, a fascinating answer. So that sets the stage for David to add his comments before we open up the discussion.
Yes, I mean, obviously my own field has been in another pathway, the P53 pathway, but I've always followed Bill and Peter's work with great interest in the whole field because of the parallels, which will definitely come out in the discussion we have. But these are really fundamentally interesting stress control pathways, and they auto-regulate in a fascinating way. And I mean, I just found I've always found it incredibly exciting to get into that kind of detail and to see, how these mechanisms evolve. And as we discuss more, we'll get into the detail, but it's incredible, the parallels. So in both cases, you have a sort of system that's kind of being turned off all the time, and then you have to do something to turn it on. And it's a very subtle protein interaction and the regulation of that interaction. And it's getting, as Peter and Bill both said, getting into the nuts and bolts of it to understand it. It really gets you into the therapeutic opportunities and the future directions. And that kind of science where you actually try to work out exactly how something works is very, very important. And I think it's getting a bit lost, in my opinion, and we'll come round to that again. But I think it's getting into the detail and pursuing a question in depth that is so much fun as a scientist, because in the end you come up with some answers. It's not just collecting observations; it's actually getting to the point where you understand enough that you can kind of intervene in interesting ways. And I think in both these systems, we've got to that point where there's molecules and there's understanding that allows intervention. So it's gone from sort of very broad general question down to something that is specific. And the type of science that we like to do is very exciting because we do get to that end game.
Yes, of course. Yes. So it's interesting that each and every one of you, in answering my opening question, have converged on a couple of points that perhaps I can challenge you on. Of course, I should raise my hands right there and say, you're singing to the choir here, but I'd like to challenge you for the sake of discussion. So one point is the importance of reductionist biology, in other words, hypothesis-driven science, as opposed to hypothesis-agnostic, larger quantitative type studies. So it's clear that reductionism has enabled us to understand mechanisms in great detail at the cellular and molecular level. But could I push back and say, well, that's all very well, but things, the way they work in cells, they don't really reflect how they work in people. So what's the point? Don't you want to understand organismal biology rather than just cellular and molecular mechanism? Any of you is welcome to answer, but perhaps I could call on Bill first.
I think stepping back, I would put scientific activities in two categories, and I think they're complementary to one another. One, as we've been talking about, is hypothesis testing, reductionist biology. The other might be more descriptive and hypothesis generating. But of course, they're complementary to one another. Different people are drawn to different types of science. I like solving puzzles, so I like to understand the mechanism. I've never seen a heat map or a Tisney plot that told me how something worked. It might have been publishable, it might have been intriguing, and it might have generated hypotheses that people like me might want to test. So the first thing I'd like to say is I acknowledge that they're complementary. I think there are times to go deep and do reductionist biology. There are times to step back and do what people might be some people call systems biology. Where I would push back a little bit on your question in terms of, well, don't we have to say whether this is what we found in the cell culture plate is true in people, for example? That's absolutely true, but I don't think that gets us out of the hypothesis testing realm. In fact, I think if you're really committed to testing your hypothesis, as you say, that is the bar to show that what you just observed in a test tube or just observed in a cell culture plate actually ever occurs in an intact animal. But I would still say that's still a form of hypothesis testing.
That's very fair, Bill. Thank you. David, what about your comments on reductionism versus organism?
Yeah, I think it's all a question of starting off from a good place, if I might put it like that. So in both in the case of P53 and the HIP pathway, we have human genetic diseases with very, very clear phenotypes that are associated with mutations in components in the pathway we're investigating. So at some level, you have a very strong, direct piece of data that tells you that this is a very good question to look at, because people who have mutations in P53 are very prone to getting cancer, and people who have mutations in VHL are very prone to getting cancer, though different sorts of propensity. But that sets you on a very firm framework. And then I think, as Bill said, I mean, once you get to a certain point, of course, you've got to test your hypothesis. So the key that's happened for both of these fields is that we've been able to generate reagents that allow you to test those hypotheses in a quantitative way. And I think that's the way that you bridge the gap. I think, it's true that you have to be careful with tissue culture, and particularly with stress responses, because, cells don't normally grow on very hard plastic surfaces in 10% fetal calf serum. So, I mean, we do have to recognize that. But I think, in both of these topics, vindication has been very strong, in fact, that those kinds of experiments have given us very meaningful answers. That's really been, is a good route to discovery. So, and of course, I guess I'm always somebody who likes to make things, and I think you make a tool that allows you to test your hypothesis. It's very, very useful. So in the case of my field, the discovery of small molecule inhibitors has been immensely powerful. I mean, it's not without its constraints, but it is very powerful because it allows you to ask questions in a very quick and specific way in many different systems.
Thank you, David. Peter, your perspective would be particularly interesting, given your role at the Crick Institute in translation of observations to the clinic.
Sure, no, thank you. I mean, it's fair to say, echoing Bill's comments, that I was not looking for a therapy when I selected this problem. I was looking for a piece of reductionist biology, actually based on the sensitivity, the extraordinary sensitivity of this response. I thought it would have a mechanism that was distinct from the countless things that arise when you make a cell severely stressed and deficient in oxygen. I thought it would be soluble, and to our good fortune, that turned out to be the case. The question is, what have we achieved? Well, we have achieved atomic scale resolution of a piece of biology. Have we solved in any way the problem of oxygen homeostasis that I alluded to in the previous answer? And the answer to that one has to be no. We have only painted one small part of that picture, and none of us can tell you how this system works with extraordinary precision in different areas of the body, with different ambient tissue oxygen tensions, for instance. All of that is not known, and it may be that the explanations are beyond our technology. We're talking about a chemical reaction, which we assume occurs just like that in cells, but it might be caged in all sorts of ways that alter its kinetics effectively. And we probably don't have the means to understand that at the moment. Now, the question of therapy arises, and again, we did not invent a therapy, we invented a target, and there is a substantial element of empiricism. I think it was not in doubt that this treatment would induce EPO levels. But what it will do to all of the other systems in the body and how that will help or hinder is an empiric question, which has now quite bravely been tested by those in the pharmaceutical industry. That empiricism is important, I think.
Thank you, Peter. So I can see from the questions that are coming from the audience, there are many people who are actually interested in some of the details of the response and what you uncovered. So perhaps turning now to Bill, could you start, Bill, by giving those in the audience who may not be experts in the hypoxia response an overview of what you consider to be the key features of the response and how it works that you, Greg, and Peter discovered?
Yes. Well, again, given the centrality of oxygen to many diseases, it's not surprising, perhaps, that once we understood the pathway in a bit more detail, there were some opportunities for new therapeutics. And so, for example, by the mid-90s, we knew that kidney cancers that had mutations in the VHL gene exhibited an insensitivity to oxygen. The oxygen sensing pathway was somehow disrupted, and these cells were acting as though they were starved of oxygen 24/7, whether oxygen was available or not. And as a consequence, we're overproducing things such as vascular endothelial growth factor, which have been implicated in tumor angiogenesis. And so we argued even back then that if the VEGF inhibitors that were fortunately being developed by the pharmaceutical industry were going to work anywhere, they would work in kidney cancer, and that's been borne out. Then over the next 5 to 10 years, through our work, Peter's work, and many others, we learned that the fundamental problem was when you lacked the VHL protein, you couldn't destroy the HIF transcription factor properly, which was a master regulator of genes such as VEGF. And of course, we and others argued that why go after a single HIF target gene? Why not go after HIF itself? And fortunately, in kidney cancer, the problematic HIF was HIF2, a lesser studied member of the family compared to the more famous canonical member HIF1. This illustrates a very important point. HIF2, 15 years ago, 10 years ago, would have been viewed as undruggable, however. But fortunately, a number of scientists were brave enough and smart enough to challenge that dogma. And two scientists who were then at UT Southwestern, Rick Pruitt and Kevin Gardner, showed that you could drug HIF2. And I must say that although it's only in phase 2 trials, actually just began phase 3 trials, the HIF2 inhibitors look very promising for kidney cancer. So that's trying to block the oxygen pathway, or at least the hijacking of the oxygen pathway in cancer. On the flip side, for diseases like anemia, heart attack, and stroke, maybe we want to go in the other direction, and now we have a first generation of compounds based on our work, and including Peter and Greg here and others in the field. Now, for the first time, we know how to pharmacologically stabilize HIF, how to turn on this program for therapeutic benefit in diseases like anemia, heart attack, and stroke. So that's been very gratifying. But as Peter said, this was all made possible by the knowledge that was generated by the field. And we were very fortunate that with that knowledge, you could start to see some opportunities.
We hear you loud and clear. All three of you have emphasized that point. David, looked at from another perspective, P53 is a sort of master regulator, if I can coin or use that old chestnut, in terms of regulating the way cells respond to stress, including hypoxia. What is it that drew you to your observations that led to the discovery of P53, and what do we know now about its role in the response to hypoxia?
Yes, I mean, it was a different route. I mean, essentially, it's a very long time ago, we were studying transformation, cancer formation in tissue culture cells. And in the pre-cloning days, and one of the great tools was a small virus, SV40 virus, and only had a 2 kilobase genome. And it basically produced a single protein, T antigen, the viral T antigen, that was responsible for this transformation phenomena. When I came into the field, the real excitement had been the production of temperature-sensitive mutants in this protein, and so using those mutants, you could show in tissue culture that you could switch the phenotype of a cell between growing normally and growing as a cancer cell by a temperature change. So that was what really got me excited. I thought it was so clear that this protein had to be central to what was happening. And so essentially we took an immunological approach to the protein. I mean, it's hilarious to think about, how difficult things were in those days. Very little, sequencing was just coming. We had no PCR reaction. We had no monoclonal antibodies. So everything, was kind of a very different stage of technology. But actually the breakthrough was setting up an assay to measure something. We measured the antibody response. We had different hamsters who got tumors, and we could measure one hamster's response versus another. In the end, we found a very strong antiserum, and that sort of allowed me to kind of purify the protein, make a monospecific antibody, polyclonal antibody. And then we just saw this protein that was coming immunoprecipitating with P50, with T antigen. And we had a lot of grief. I mean, basically at that point, everybody said, oh, it's a fragment, it's a broken, piece of T antigen, it's a background band. It does run, exactly where the immunoglobulin heavy chain runs. So, I mean, one of the reflections is this sort of technical thing that you have to get into to make a discovery. I mean, why we were successful was lots of small technical innovations. We just started to use protein A as heat-killed bacteria. That cleaned up immunoprecipitation enormously. We made this antibody that was, about 1,000 times more potent than what we had before, and suddenly you can see things, and then you have to take the leap, which is to say, on the basis of my relatively weak data, I'm convinced this is a host protein, not a viral protein, and publish with a bold title. But I mean, it was, it was very interesting. I mean, I think we all forget sort of what things were like at the moment, and everything becomes so sort of accepted. But when you do science, it's not like that at all. I remember when the paper came out, my colleague at Cold Spring Harbor at the time told me I'd made a complete fool of myself, and it was obviously an artifact, and that was his response to the paper. So I think sometimes when young people kind of start in science, they get this sense of it all being sort of like very straightforward, delivered knowledge, but it isn't. It's a constant struggle of process. And it's this, I think, the balance that both Bill and Peter have talked about, where you have to sort of balance your cynicism versus your optimism and kind of find the right place. And
I think it's called rigor, perhaps. Yes.
Yeah, it's a mixture of the two. If you don't get excited, you don't get anywhere. So I think, one of the characteristics of being a scientist is sort of getting excited and then finding out that you're wrong, and dealing with it and picking yourself up, going on again. So you do need to be optimistic. I mean, just yesterday, I found from a colleague that something I thought he'd discovered of extraordinary interest, and he just told me, actually, no, it's gone away. And this is quite a common experience, actually. So I think, but the good thing about it is that, the great thing about science is you will get there in the end. I mean, there is a certain reality there that you can get to. So I guess that's what the puzzle solvers among us like that.
Absolutely. Thank you, David, for that exposition. Peter, turning to you, what was it that led you to think about the hypoxia or the cellular oxygen sensing mechanism? Was it, as Bill was talking about, an interest in understanding a particular disease associated with cancer? Was it just a pure curiosity about how cells sense oxygen?
Ashok, I think in fairness, it was curiosity. As I say, I selected this as a problem that I thought was capable of solution. I was hopelessly overconfident at the time and thought that I could solve it, which in fairness, I did contribute to the solution. But had any rational person assessed the likelihood of that at the time, that they would have drawn a negative conclusion. I started in 1988 and through the 80s had thrashed around thinking of what I might do. And it's just interesting to hear what David said. At some stage during that time, I became very irritated by my so-called mentors, because it became obvious to me that those people studying small viruses that cause cancer would find out important things. And I concluded almost that people had been obscuring this from my vision because it was so downright obvious. They should have known and should have told me. I came into this from clinical medicine. I don't have a scientific training at all, really. On reflection, though, obviously I was too late. That had become obvious to others beforehand, and actually I was lucky that they hadn't told me about it, because they would have brought me into a crowded field, and we were better off where we were. But for the young people listening, actually I think these ideas are obvious once you've had them. And we actually had, from maybe 2 years into the work, I had little doubt that we were onto something interesting. Other people had doubts at that stage, but we didn't. And it's a famous paper that was rejected by Nature.
Yes.
And, we've brought it out into the light now, but be sure, we had the arrogance to put that paper, that rejection letter aside at the time, and thought it might come in handy and interesting in the future. We put in 1992, it went in Chris Pugh's drawer, and he fished it out for a particular reason in October 2019. So that's the lesson to the young people. These ideas are downright obvious once they've occurred to you, but everyone else will confuse you. They will try to get you to do other things that they think are important, and you just have to hold your mind and have the confidence.
Absolutely. I think there are some very valuable lessons in what you've said, and Bill would like to chip in.
Just a brief follow-up, because I think if we're going to extol the virtues of reductionist biology, we should give our listeners maybe a couple hints of how to get there. And in listening to David and Peter, and I felt this for quite some time. One of the problems with purely descriptive science is you wind up doing what I call the molecular equivalent of the blind man and the elephant.
Yes.
Where everyone's measuring things, they're all true, but you don't know what's causal and what's correlative. And of course, the great tool to establish causality is genetics. So P53 mutations told you unequivocally P53 was important in cancer. It turns out it's important for many other things, but it unequivocally was important in cancer. VHL mutations told us that the VHL gene was unequivocally important in kidney cancer, and the clinical findings in patients who carried VHL mutations told us it was almost certainly involved in oxygen sensing as well, et cetera, for HIF. So I think having a genetic anchor to start with is often a very good place. And if you can marry that to biochemistry, which gives you the tools of mechanistic understanding, then you're off to the races.
No, I think that's a very interesting insight, how different fields can come together to really get you to the bottom of a mechanism. So all three of you alluded in different ways to cellular responses to stress, in this case hypoxia, or in the case of P53 that David was talking about far more generally. So could we turn now to the problem of cancer and carcinogenesis? Bill, perhaps you could tell us about how the cellular response to hypoxia, when it's inhibited, actually promotes carcinogenesis. You alluded to it concerning VHL, but perhaps you could tell us a little bit more. There are many questions about the detail amongst the audience.
Yeah. Well, first of all, at a very high altitude, as we all, I suspect, agree, learning about cancer pathophysiology teaches us about normal physiology and vice versa. It just so happened that the VHL gene was particularly powerful for understanding the oxygen response. And acquiring enough oxygen is one of only many challenges a tumor faces as it's expanding and outgrowing its blood supply. So it's been appreciated back to at least the 19th century that one of the challenges for tumors is to obtain a blood supply, presumably to obtain oxygen and other nutrients. Now, for the first time, we understand one of the ways this can happen. The tumor cell can hijack this oxygen sensing pathway, rev up HIF, and as a result, turn on things like VEGF. Now, the dirty little secret in cancer biology, and I'm what's called a lumper rather than a splitter. So I'd like it to be true if once you understood the rule, it was always the rule and it was the rule everywhere. But for reasons we don't completely understand, different cancers arising in different tissues sometimes solve these problems in different ways.
Yep.
And there are even examples where a given protein might be pro-tumorigenic in one context and actually anti-tumorigenic in another. So sometimes you actually do have to be a splitter rather than a lumper. We're still learning why kidney cancer in particular is so dependent on HIF, and in particular HIF2. But I think to some extent, all cancers have to solve this problem of getting enough oxygen, getting enough blood, put it another way.
Thank you, Bill, for that interesting perspective. Over to David now. I mean, the gene, the protein that you discovered, as you described, David, has turned out to be one of the most important, if not the most important protein involved in suppressing tumorigenesis in humans. How were those connections elicited? What role, why is P53 so important in stress responses, and how does its alteration contribute to carcinogenesis?
Yes, I mean, it's still, I think a good dose of humility here. I think we don't by any means fully understand. We had a lovely period in the, late 90s when we thought we had it pretty understood, because we had a very nice pathway by which DNA damage could induce the stability and activity of P53. And then P53 turned on genes that induced apoptosis and cell cycle arrest. And it was very, very nice. We did experiments to test this very beautiful hypothesis, and things became more complicated. So you could knock out the genes that are the primary targets inducing apoptosis and inducing cell cycle arrest, and still get very good tumor suppression by P53. And so the fundamentals are very clear that this pathway is extremely critical in regulating genetic stability. It's very critical in regulating aspects of metabolism. It's involved in control of aspects of cell motility and invasion. So a very broad spectrum of activities. But which one of these is responsible for tumor suppression is less clear. Is it a combination of all of them? We know that, a bit like HIF, that there's a large group of target genes. So P53 can activate the transcription of a core of at least 100 genes. And the regulation is very complex. So in some cells, you'll see some particular genes being activated by P53, and in others, you'll see others. So there isn't an absolutely universal, answer to the transcriptional response. And so I think, this is where reductionism becomes challenging, because you have a complexity of response. And the question is, can you pin it down, or are we looking at a phenomena that is sort of very redundant and overlapping? So there's a limit to how reductionist we can be. And I think that's, the field is kind of very much engaged in that kind of study at the moment. And typically in any field, you'll see scientists with wildly different views. And so one of the things that makes it exciting to work in is to see the variety of different opinions. And I think, there are these clues that look very exciting, but they can also be misleading. So one of the features of the P53 system is we see the accumulation of the protein, usually the mutant protein in tumors. But that's a very interesting observation, but how significant it is biologically is still very controversial. And I think still, under much discussion. So the key mechanism of P53 does seem to be this stress-induced activation, which then has this pleiotropic effect, which combine to seem to limit cancer. But to come back on the humble, we do know that about half of human cancers don't mutate this gene, and what does that mean? Is there a completely parallel pathway that is equally important that we haven't quite discovered, or is it that all these other tumors have found another way to block the pathway? And just to be a bit sort of philosophical, I think one of the issues that we have as scientists is the systems that we study. So when we study what we call normal P53 responses, we often study those in cancer cells that have actually in some way bypassed the system because it's convenient to do so. So, recently we've started to work a lot more on the response in whole animal species and look very carefully at the more systemic nature of the response, because I think, that's an area that we have to understand.
Thank you.
Especially when drugs come in.
No, I absolutely, and I'm going to turn to that right now, because I'm going to ask Peter to comment on something that you had alluded to, and he had too earlier, which is this notion of pleiotropy, tissue specificity, and which you alluded to, David, and what Peter talked about earlier, about the way cells can set thresholds by caging or kinetics. Peter, when you are trying to translate fundamental discoveries of the kind that all three of you have made to therapies, how important are these types of issues and understanding these types of issues?
Well, they're totally key, Ashok. But whether we can understand them or simply deal with them empirically is a question, but there's no question as to their importance. Now, I notice a lot of people have asked questions about cancer, and you've been discussing cancer. So I'll give you a slightly personal perspective on cancer as a non-oncologist. It goes like this. I was asked to give a lecture in memory of Sir William Osler, and I thought I'd talk about cancer. That was an arrogant thing, because I'm not an oncologist and know little about the disease. It was also ignorant of Osler's life and works, because the one thing he didn't write a lot about was cancer. He actually believed there might be a psychological cause at some time, but it drew me into going back to the turn of the 20th century and the evolution of thinking on cancer. So they were perplexed by four observations, really: that X-rays cause cancer, that chemicals cause cancer, that viruses cause cancer, Rau's sarcoma virus, and that you could inherit cancer. And this seemed extremely puzzling, and of course was resolved in the 1960s after the structure of DNA and mutagenesis assays were perfected, when it became clear that all of these things were mutagens and could alter DNA in some way or other, or reflected altered DNA. So there then appeared to be a beautiful synthesis, just as David alluded to with P53. We thought we had a moment of truth, that cancer was straightforwardly a disease of DNA and mutation, and there you go. And then the interesting thing was that people began to identify the specific genes that caused cancer through inheritance or whatever. And that has proved extremely interesting, that the genes have been identified and they have, in the main, critical functions. But what's become apparent is that only very specific mutations in those genes cause cancer, number one. And despite their general action in biology, they usually only cause
tissue-specific
types of cancer. So that specificity is alarming. It's stunning, and it is completely in opposition to the original idea that these genes were cancer-causing genes. David's system, P53, is about the only one that shows reasonably general actions. So this is an extraordinary puzzle as to why it could possibly be that cancer is mutationally constrained and context constrained to quite such a remarkable degree. So we have hit upon the idea, based on our work on hypoxia pathways, that the only credible explanation for this is massive interconnectivity of pathways. And this is the difference between, as we said, understanding oxygen sensing at the level of a reductionist biology, and then could we infer from that how it worked in oxygen homeostasis. That combination, connectivity, is truly mind-blowing, I believe, and that in some way, not yet understood, it explains these remarkably specific effects that we are seeing. And I leave it open there. I mean, one technical thing, I think we're seeing a lot of epigenetic regulators that are mutated in cancer, and I really don't know why. I really do not know why. The evidence, genetic evidence that they're important is crystal clear, but I'm not really persuaded by the idea they just nudge the system one way or another. I don't think that's credible, but I do think it's connected with this extraordinary specificity of cancer. As Ian Tomlinson put it, everything has to be just right to get cancer, just right. And I don't understand what the reductionist biology of just right is, and I don't understand those epigenetic regulators either. So that's my speech
Peter.
on cancer as someone who has never practiced oncology and never came into the field wanting to do oncology.
But nevertheless, I think there are some really tremendous insights in what you, Bill, and David have said. So I'm going to turn now, because your discussion has actually provoked a number of interesting questions from the audience. So I'm going to call on one or two people. I'll get them to state their questions, and please, any of the three of you who'd like to sort of answer, please go ahead. If we keep the answers fairly brief, then we'll have time to go to more questions. So some of the questions that I would like to start with actually pertain to what we have just been discussing in the last few minutes. And one is from Adviti Nayak at the Qatar Biomedical Research Institute. Adviti, are you online? Could you convey your question, please?
Hello, can you hear me?
Yeah. Fire away.
Firstly, it's an honor to be on this panel asking this question. So my question to you is, since you alluded to the great interactability between all these different pathways, could you comment on mechanisms of interaction between the adaptation to hypoxia in cancer cells and the DNA damage response, since you're also talking about P53, because it seems to be quite central in modulating such diverse effects in cancer cells. So do you have any comments or opinions on this? Thank you.
That's a difficult question. You've stunned them into silence.
Well, there's no doubt that there's tremendous crosstalk between the hypoxia pathway and the DNA damage pathway. This shouldn't be surprising, because I figured out a long time ago everything's connected, right? So it gets back to the blind man and the elephant. It's all connected, and so I think I don't think we need to go into great detail, but there's certainly examples where HIF can affect the P53 response and vice versa, and HIF at least indirectly controls some genes involved in the DNA damage response. But I think, I always step back and say, what question are you trying to answer? And does the question sound interesting and important if you say it out loud? Or put another way, can this question be reduced to something which some people would call tractable, meaning I could break this down into an experiment I could actually understand? I mean, one of the things that's helped me is that I'm not, super smart, so I gravitate towards experiments that are simple enough. I think I actually will understand their execution and their interpretation. So that's another way of looking at it. Can I break this question down to an experiment that sounds like it's doable and that I would be able to interpret. So I would need to know more about what interests you in this question, I suppose.
Yes, I mean, I think you know, I had a quick look at the literature prior to this this session on the crosstalk between hypoxia and P53, and the literature is deeply confusing. So it's an example of this problem of sort of collecting information without sort of focusing down, as Bill says, on a particular question. So I think one of the bits that's clearer is all about angiogenesis. So I think because the evidence that P53 can suppress angiogenesis is quite good, and the evidence that that angiogenesis is driven through molecules triggered by the HIF pathway is good. So I would look there and try and understand the crosstalk between the two pathways in a rather simpler system, because at the moment the you know, the literature is very confused. Some people say that you know, there's a very strong P53 response to hypoxia. Other people say there isn't. I guess the other thing to look for is points of sensitivity. So a very good example of that is P300. It's a transcription factor that's required to recruit RNA polymerase to the start site to allow transcription, and it's haploinsufficient. That means that the cell doesn't make all that much of it. And a variety of transcription factors all need this cofactor. So when you have a cell and you start making a lot of P53, it can sequester P300 from HIF and vice versa. So again, you want to look at these potential points of sensitivity, and genetics is very important in that approach. So this, if we find a gene that's haploinsufficient, that it means that the cell can tell whether it's got one or two copies of the gene. That's a good place to start.
Thank you, David. Before I turn to Peter, I'm actually going to bring in someone from the audience, Bertie Ansell from the UK, who had a question on this topic, which perhaps Peter would care to address.
The same topic, Asha.
Very same topic about how does one begin to tease out Bertie. I'm just waiting for him to come online. No? Okay. So I'm just going to read out the question, which is, how close are we to understanding integrating detailed mechanisms of cancer pathology across distinct signaling pathways, and how can we tease out cross-pathway interactions? So very much along the lines that you've been discussing.
Yeah. Well, a difficult one, really. You're just sort of echoing back on I think, Bill's answer. The complexity is such that everything is connected to everything else. I don't actually like crosstalk papers in the literature. I now won't review them. And I believe them, as David has alluded to, be almost universally incorrect. Now, what is the reason for that? And it is that because of the connectivity, it is extremely easy to confuse direct and indirect interaction. And of course, the indirect interactions will be context specific and hence not reproducible in another context, and we're seeing hundreds or thousands of examples of that, usually in the high-end literature, the Cell journal stuff, and it's a really difficult problem. And I think the answer is that in order to prove a direct interaction or a direct piece of connectivity which holds good in multiple circumstances, first of all, the standard of proof should be very high. The second, it should involve a biochemical element, in my view. And third, it should be tested in multiple contexts. And if you do that, you will tease out the direct from the indirect. But think of the maths. If every system has 10 direct interactions, then by the time we're on the fifth order, we're at 100,000, of course. So that those indirect things massively outnumber the direct things, and therefore the possibility of error is magnified.
Excellent.
I hope that wasn't reveal undue cynicism, but just be really careful about pathway interaction stuff. As David says, I don't know how many papers have been done on HIF and P53, and actually you can't review it. It's not possible to make any synthesis, to my knowledge, of what's been discovered.
Thank you, Peter. That's very clear, and I think it does address some of the complexities that the audience have been raising. In the last few minutes, let me turn to a question on a different topic and perhaps ask Bill to kick off, because he spoke to it earlier. And this is from Juanita Klingam in Thailand. Is she ready to come online? It's about HIF1 inhibitors and therapy.
Hello, do you hear me?
Yes.
Okay, I'm glad that to have a chance to talk with you guys. So my question is, are any specific HIF1 inhibitors or any another related protein that's involved with HIF1 pathway that is already approved in the clinical studies? And if not, what you guys think was the reason that the drug cannot go to the clinical studies?
Juanita, could you please mute when you're not talking because we're getting some background? Bill, the question was there...
Great question.
Yeah, you've got it.
Here. So great question. So first of all, as I mentioned, despite the naysayers, some very clever structural biologists helped us develop the first generation HIF2 inhibitor. There's a pocket in HIF2 alpha that when bound to a drug undergoes an allosteric change that prevents HIF2 from binding to DNA. Unfortunately, the equivalent pocket in HIF1 alpha is said to be too small to accommodate a drug. So it's been much more difficult to drug HIF1, although Peter's colleague, Freydun Rastinajad, is one of the people, I think, leading the field in this regard. But I'll make one editorial comment, which is there are nonetheless in the literature quite a few HIF1 inhibitors. But if you really look at them, they're subject to the same artifact, I'm afraid, and that is HIF1 turns over pretty rapidly, certainly compared to the usual loading control like actin or tubulin. So if you poison a cell, you can be sure HIF1 will disappear before the loading control. So what I tell my students is the least interesting explanation for your data is the explanation until you prove otherwise. So if you read a paper with a HIF1 agonist, and it looks like HIF1 is going away, until proven otherwise, it is a toxin.
Thank you, Bill. In the last few minutes that we have, I'd like to invite one last question from Helen Rodriguez-Caro at the University of Oxford, so very close to Peter. She's asked a number of questions about the passion for science and how one follows a career. And I'll leave it to her to actually formulate her question. Nope, she's offline, I'm afraid. So she wondered about how much do you all think that the reductionist approaches that you have taken in your careers and you've eloquently spoken to will continue to play an important role in biomedical science? And I think that's a suitably philosophical question for us to end on. So very briefly, please. David, perhaps to start with?
Well, I'm pretty passionate about this. I feel that it's vital that we continue to do this kind of work. And I think where science has gone wrong, actually, is not asking this degree of precision and reproducibility. And I mean, Bill's written very eloquently about it, actually, the idea that you have to build on solid bricks. And so it should be perfectly good enough to publish a paper that simply determines something very accurately. And I think a lot of the issues that we face now is a huge amount of data being collected, but not really being analyzed. And I don't want to go on too long, but it's one of the key disciplines is to bring together genetics and biochemistry. And that's the way that you can find things out. And we've lost biochemistry, and we need to get it back. And if you look carefully at what Bill and Peter have done, at its core is biochemistry, protein chemistry. That's how these answers were found, and it's very much the same in the P53 pathway. But the number of people studying protein chemistry these days is rather restricted.
Yeah.
Point taken. Thank you, David. Over to Bill, and then very briefly to Peter, and then we'll have to wind up.
Yeah, I love this question on multiple levels. So the first thing I would say is, find something you're interesting and passionate about. I mean, what makes the world go around is different people find different things interesting. So please find something that you yourself find interesting and you're really passionate about it. And you should really feel that some of science is fun. Okay, there's some headaches once in a while, but most of the time it's actually a great privilege because it's just so much fun. Secondly, I think you have to examine your own temperament. I mean, some people are temperamentally better to be scientists, others are better temperamentally to be engineers. So if getting an unexpected result is categorically going to destroy you, emotionally and otherwise, maybe you want to be an engineer where you live for things just working the way they're supposed to work. A great scientist actually, I think, would be disappointed if every single experiment worked exactly the way they thought it was going to work. I think it was Isaac Asimov who said that most great discoveries actually start with someone saying, "Wow, that's funny. That's I didn't expect that." That's certainly true. So I think you have to understand what your tolerance is for the unexpected result, and whether you're courageous enough to follow the unexpected result. But first and foremost, find something you're interested and passionate about, and then, as David said, go deep.
Thank you, Bill. And Peter has the last word from the panel.
Well, finally, yes. The passion is the key. All sorts of words have been used to describe this type of science. Blue skies, I mean, that sounds a casual. Curiosity, also casual. Bottom up, that's just plain boring. Passion best describes it, and you should be passionate. There is just one rider to that, which Bill has alluded to. Most passionate people are intolerant of evidence that's adverse to their belief. So you have to be able to be tolerant and passionate. Otherwise, you may be unhappy. So please be tolerant and passionate and therefore happy with your science.
Thank you very much, Peter. And that is about time for us to conclude. Let me thank you all very warmly once again. It's been such a pleasure and a privilege to talk with you informally in this setting. And I know that I, as well as the audience, have got a lot out of it. And particularly your answers to the last question, I think, are a sort of ray of sunshine in the grim COVID epidemic that we're facing worldwide. And I hope it'll inspire a lot of people, a lot more people, to do science. So Bill, Peter, David, thank you all very much once again. And good night to you from Singapore. Goodbye.
Thank you. Thank you.
Thank you.